Before I get into some of the detailed projects, I always like to set the stage just to make sure everybody's familiar with epidemiology. Occasionally people think I study skin disease, and I don't really want to look at any weird rashes you might have, but what it really is looking at diseases over a population level, and it's based on some of the dynamics between associations with the host, the pathogen, and the environment. And that kind of relationship can be used to look at disease over time and space, as well as develop risk factor models of who gets disease and why. And all these methodologies were developed actually to study human disease. But when we talk about applying this to a marine mammal population in a very dynamic environment that Dennis kind of mentioned, it turns into a bit of an ADD nightmare here. And the details of this really aren't that important, but I want to get across the concept that there's a lot of complex interactions going on in Lagoon that ultimately affect health, not just the health of the dolphins, but also feedback to the ecosystem and potentially human health. There's a lot of inputs, a lot of data that's needed. The observatory is actually a good source for some of the environmental data that can all be tied into this. And so again, I just want you to get the concept that there's not necessarily one smoking gun to explain all the things that we see with dolphin health. It's really kind of a combination of a lot of factors, which keeps me plenty busy. But it also allows us to understand things at a little bit wider level than just simply dolphin health, but using them as a sentinel or an indicator species for specific diseases or contaminants. Again, it can help us answer some questions about the ecosystem. And then we've been able to develop some other projects that are actually looking at the risk to coastal human populations as well. So to get into a few details, this was some work that was published a little while back looking at a variety of infectious and potential zoological diseases. And I just want to pull out a couple that, again, have some environmental impacts here with toxoplasma. This can also be a human pathogen affecting pregnant females. And what's interesting about it is the cycle here. You probably can't see that, but that's supposed to be a cat. These are whatever it's supposed to be. They can shed the pathogen into the environment, and we've actually seen dolphins with evidence of exposure to toxo. And a Link intern last summer started to look at some potential environmental inputs and risk factors associated with that. And then West Nile virus got a lot of attention a few years back. It's the first time it's been reported in a cetacean population where dolphins have been exposed. The primary host, the reservoir, are birds. Then you have the vector. Humans, horses, and even dolphins can get bit by mosquitoes. And then an interesting disease that we've been doing a lot of work on, this is some collaborative work with Peter McCarthy and Esther Guzman as well, is lobomycosis. And it's a dermatophyte that causes these really nasty kind of cauliflower-like lesions on the dolphins. And it's only been observed in dolphin populations and humans, but it's primarily endemic to South America in the human cases, but we see it mostly in the southern IRL with the dolphins. The really interesting thing is that it's never been grown in culture before, the actual causative agent. So there's a lot of unknowns, questions that need to be answered. So we started looking at some basic and applied research where we're trying to grow it in culture. And to do that, simple methodology of just taking the lesions and preserving them to keep the cells viable, determining if they're viable using some live dead staining, and then simply isolating the causative agent from the dolphin bits. And that's here. It's kind of hard to see, but I promise you those are actual Lacasia cells, and that's just some junk behind it. And by doing that, we were able to inoculate a variety of both traditional and novel microbiology media, including epidermal cell cultures and some skin graft material. We've been able to work with some physicians in Vero to get human skin graft material to try to inoculate and get this fungus to grow. And then some molecular work that's going to allow us to look at the similarity of the strain with the dolphin disease and humans, and also hopefully develop a molecular probe where we can actually go out into the environment and try to detect the presence of this fungus to, again, try to find some sources of it. Contaminant work has been a big one as well. We just had a paper that was published in Environmental Toxicology and Chemistry on PFCs. It was the first time we've been able to look at some of the potential pathophysiological effects of high-end exposure to PFCs in bottlenose dolphins. And we were able to tie that in, and we're developing some projects where we're looking at co-exposures to a variety of chemical contaminants. Because again, the lagoon is a very dynamic place, and these dolphins aren't just exposed to one contaminant or one metal at any given time. So we're continuing to do surveillance and develop that work. The other heavy metal that a lot of research has been done is in mercury, and again, looking at some of the basic physiological parameters, and we've seen evidence of hepatic, renal, and even endocrine dysfunction where they're not producing some thyroid hormones as readily when they're exposed to high concentrations of mercury. And the immune component, which is really interesting. And basically, it suggested that these animals have a reduced capacity to mount an immune response when they're exposed to a variety of pathogens. And that's all associated with high-end mercury exposure. But again, we're trying to tie that into some of the other chemical contaminant exposure as well. And also done some basic spatial and temporal work to, again, look for potential hotspots in the lagoon. And this SLP work has also allowed us to kind of leverage it to develop some other projects that aren't SLP funded but are quite interesting. One of them is looking at gene expression associated with mercury exposure. That's working with Sarah Edge, Lisa Cohen, and Steve Field. We've actually seen concentrations of mercury in these dolphins that in the lagoon that are some of the highest that have ever been reported in cetaceans. So that led to some questions about what's going on in the food web beneath it. So I was able to get some funding from the Georgia Aquarium to sample selected dolphin prey species to look at trophic transfer and bioaccumulation in the environment. In that paper, I just finished edits on that draft yesterday, so hopefully it'll be submitted soon. And then this really cool work, first of its kind, to look at shared environmental risk with humans. So we went out for 2 summers with another Link intern and sampled recreational anglers to determine what they're catching and what they're consuming and how often from the lagoon, and tying that into their hair mercury concentration. And that paper actually is in draft 2. And that Link intern is presenting her undergraduate thesis today for her graduation requirement, actually. And antibiotic-resistant organisms, this will tie nicely into what Peter's going to talk about, but we've seen a variety of potential pathogens that have been cultured from the dolphins during the HERA project. And some of these organisms have shown resistance to antibiotics that are commonly used in veterinary and human medicine, which are things you wouldn't necessarily expect to see. And some of the overall patterns have some similarity to the patterns of resistance that you would expect to find in cultures from a human hospital. And the big question is, where are these coming from? Is it direct transfer of resistant organisms to the lagoon? Or are antibiotics getting into the lagoon and driving selective pressure? So there's a couple ways to answer that. First of all, dipping into the molecular pod again, looking at similarity of strains between dolphin, humans, some agricultural, environmental isolates, and looking at risk factors for colonization with specific pathogens. And so I mentioned doing this risk factor work, and a recent paper that we did that kind of speaks to integrating multiple streams of data was looking at colonization with antibiotic-resistant E. coli, which is a great fecal indicator bacteria. So we're able to look at the spatial distribution of the cases, and this is working with Marilyn in the photo ID group. We're able to get an idea of where these individual dolphins are hanging out and kind of weight that average of their sighting history to tie it into anthropogenic and other risk factors. So we looked, again, spatial distribution. This is actually human population density along the lagoon. These are sites for sewage treatment, and even looking at density of septic tanks, which was actually a significant risk factor. And then looking at it temporally, we saw a peak in colonization of resistant E. coli among the dolphins sampled in 2006 around those storm events, which does make sense considering the environmental impact. There are a ton of collaborators and a lot of people who have contributed to this work and continue to contribute because, again, it's connecting multiple streams of data. So we have our Harbor Branch team as well as Georgia Aquarium and all of our funding sources. And with that, I think I'm right on time, right?